Refactoring photosynthesis isn't something that's been done yet. However, I want you to understand what goes on inside the more complex gene clusters to see the diversity of processes that can occur. There are many advanced gene clusters in prokaryotes. Magnetosomes are another really popular example. In terms of us having extensive knowledge about what is encoded by these clusters in molecular terms, no process is better studied than photosynthesis. Thus, I will describe to you how this process works and how you might approach refactoring it. Refactoring photosystems is one of the problems on the 10-year horizon for our field, but is impractical with the existing infrastructure. Nevertheless, it is a good example for thinking through how to approach designing biology in a modular sort of way. Many bacteria, including the cyanobacteria, perform photosynthesis. The overall process is split into two portions: the light harvesting system, otherwise known as the photosystem, and the carbon fixation process, that is called a carboxysome. Both of these processes are in some way compartmentalized within the bacteria. The carboxysome is an array of enzymes inside a proteinaceous shell, and the photosystem is a series of chromophores decorating a large integral membrane protein. This system has been studied extensively from a genetics and biochemistry perspective. The importance and role of the many genes is well known. However, when you move the gene cluster into E. coli, nothing happens. Thus, making a photosynthetic device for use in E. coli or other organisms would require a refactoring effort. If we want to reconstruct photosynthesis in E. coli, we're going to need to simplify the problem. There are hundreds of kb of genes involved in such an experiment, so we'll need to be able to debug the system with simpler substructure of the entire process. A clear modularity point in breaking this apart is between the photosystem and the carboxysome. Genetic analysis in bacteria containing these clusters shows that the connectivity between the two processes is simply the transfer of metabolic intermediates. For example, the photosystem process continues to occur even when there is a knockout of the carboxysome, so they aren't functionally dependent upon one another. So we should be able to separately engineer the two processes. We can separate the task into making distinct carbon fixing and light harvesting devices. Fortunately, all the genes for photosynthesis are known and extensively studied with plenty of genetics. Furthermore, all the enzymes needed to do the Calvin cycle are housed within the carboxysome and are similarly well studied. The first one to tackle is the photosystem. The most studied gene cluster for a photosystem is from Rhodobacter spheroides. Most organisms have two such photosystems, PS1 and PS2, and we'll focus on PS1, though PS2 is a fairly similar process. PS1 encodes two metabolites which are pigments. One is a carotenoid and the other a bacteriochlorophyll. It also encodes a multi-protein scaffold that holds these pigments in place in the inner membrane. Light absorbed by the chromophores is translated into chemical energy by splitting water-releasing oxygen and protons. This results in a proton gradient. Proton influx drives ATP synthase, which generates ATP. Thus, the photosystem converts light energy into the chemical energy stored in phosphodiester bonds. The gene cluster encoding photosystem 1 is 40.7 kb in length and contains 38 open reading frames. It is divided into around 8 operons with a complicated regulatory relay under the control of a transcription factor called CRTJ. Since we are going to refactor this thing, we will ignore all this regulation. Thus, we just need to focus on the coding sequences. Within the photosystem, there are multiple distinct processes underway. There are 3 distinct operons, each encoding monofunctional enzyme pathways for production of the pigments. There are operons encoding each component of the protein scaffold, and then additional genes encode related processes such as urea metabolism. If we want to refactor this, we probably need to break it down further into more lab-manageable chunks. The entire gene cluster encodes a singular process in the sense that if we enumerated all the intramolecular interactions in this system, we'd find that each gene touches another gene either by interactions of the proteins in a complex or through the sharing of a metabolite. The most practical way to decompose a project like this would be to divide the task by means of the smaller processes that have assayable phenotypes. One clear place to do this would be around the biosynthetic operons. Though the pigment product of these operons becomes incorporated into the photosystem, the biosynthesis of the pigment itself would occur regardless of whether the photosystem is present or not. Thus, we should be able to construct operons that just make the pigments as a first step towards constructing the entire device. Carotenoid biosynthetic pathways have been extensively explored. Like all terpenoids, they fan out from IPP, and more specifically from geranyl-geranyl diphosphate and then phytoene. Two common carotenoids in photosystems are spheroidenone and hydroxyspheroidenone, and both are biosynthetically reachable using normal monofunctional enzymes. Thus, the task of producing this substructure of the photosystem is no different than any other small molecule biosynthesis problem. Bacteriochlorophyll biosynthesis is similarly just a small molecule biosynthesis problem. It has been noted that it is a very challenging one because these pigments tend to be toxic when there is no light harvesting complex to absorb the product. Also, it is a particularly long biosynthesis, which would make it more challenging, but nevertheless, it's essentially a small molecule biosynthesis problem. Protoporphyrin IX is a key intermediate in the biosynthesis of bacteriochlorophyll, but it is also the penultimate step of heme biosynthesis. Thus, most organisms, including E. coli and yeast, already make this intermediate. The remainder of the genes are specific to photosynthesis and are encoded within the PS1 gene cluster. In addition to producing all the pigments, they must be taken up into the light harvesting complex, which also must be expressed. Fortunately, it is known you can functionally separate these steps. If you knock out the biosynthesis of these pigments in the source organism, they can be complemented by exogenous feeding of the pigments with restoration of function. Thus, constructing the photosystem could be divided into three parallel tracks: refactoring of the scaffolding proteins, refactoring of bacteriochlorophyll, and refactoring of the carotenoid biosynthesis.